All right, so I work for the Botanic Gardens of Sydney, which is made up of 3 botanic gardens, one on the harbour on Gadigal country, one in the Blue Mountains on Darug country, and one out in southwest Sydney, which is where I work on Dharawal country. So I'd just like to extend my respects to the elders of all those nations and to any First Nations people here today. So out where I work on the Australian Botanic Garden, we have the Australian Plant Bank, and that's a conservation and research facility to support in situ and ex situ conservation. So we have facilities for seed banking, facilities for tissue culture, and also for cryopreservation and generating and maintaining living collections. And we've been putting all of those to use to try and handle the myrtle rust problem because, we didn't know how species were going to react. We were running out of seeds in the wild, and so we were just throwing everything at it that we had. But today I want to talk to you particularly about seed banking. And, there we go. Seed banking is the most efficient ex situ conservation method. So you can store a lot of diversity in a very small amount of space, and it takes very little maintenance once you have them in storage. Seed banking is really appropriate for a lot of the species in the Myrtaceae, so all of those that come from drier climates, especially that produce dry fruits. And they have seeds that will tolerate drying down to a low moisture content and storage at minus 20 degrees, which is all you really need to be able to be seed banked. So that encompasses quite a few of our Myrtaceae species, so Angophora, Eucalyptus, Corymbia, Leptospermum, Melaleuca, all those species that produce small seeds from dry fruits are really suitable for seed banking. Myrtaceae from wetter environments are a bit more complex. So we do have some species from, say, rainforests, wet sclerophyll forests and swamplands that do produce orthodox seeds, so some Melaleucas, some Leptospermum, Lophostomen and Syncarpia, for example. But then we have this other mixed bag of species. So we have recalcitrant species, which includes Eugenia and most syzygium species. They'll produce fleshy fruits and seeds that don't tolerate any drying whatsoever. So we can't store those in the seed bank, and we have to try and preserve them either with living collections or in tissue culture. And Lyndall's going to talk some more about that in the next talk. And then we have this other mixed bag of species that fall in a category we sort of give umbrella term intermediate, but that includes some species that won't tolerate as much drying as the orthodox species, or they'll tolerate drying but they won't tolerate freezing, or they'll be really short-lived in storage. And quite a few of those species in the Myrtaceae family, those sort of behaviours are common amongst the species in the Myrtaceae that produce fleshy fruits. The small ones, sort of like the berries, so they've got small fruits, they're fleshy, and lots of seeds inside, and they're showing this strange intermediate behaviour which is making our lives a little bit difficult. And we didn't realise this was such a problem until about 2015, when we pulled some seeds out that we'd had in storage, and we were just testing them to make sure they were still viable before we did some other experiments. And we found this horrible thing, and it's just one of those moments where you go, it's not what we wanted. So these species were dried, they were tested after drying, they germinated beautifully. We put them into storage thinking they'd be fine, and then within 3 years we've pulled them out, and storage has declined to 0 for some and to less than 20% for others. On the right there you can see what's happening with Rhodomyrtus sidioides. So we did a comparison here. We had some control seeds stored at 15 degrees and another packet of seeds stored at minus 20, and the ones stored at minus 20 you can see not producing good roots, not producing good shoots in comparison to the ones that were just stored at 15 degrees. So that's a problem, because when you're seed banking you want to try and store the seeds as cold as possible and as dry as possible to maintain their longevity, and that's been our standard operating basis for everything that we've been working with. And for most species in Australia, that's a really good way to bank the seeds, but it's turning out not to be the case for these Myrtaceae species. So we started off on a program funded by the Ian Potter Foundation to try and find out what the problem was with these seeds and why they were responding this way. So basically we decided to use thermal analysis to see what was happening inside the seeds as they were freezing and thawing. And this is using a fancy piece of kit that we got a few years ago called the differential scanning calorimeter. You put a small sample of the seed in a little aluminium pan, and you have a sample, a reference pan that has nothing in it. They get sit into this machine, and the temperature gets taken down to minus 150 and then raised back up to 50 degrees. And the machine records differences between those two pans in the uptake and the release of energy. And then that's shown up on a chart as peaks that are related to the temperature that the transitions are happening at. So just as an example, this is a species that we know was short-lived in storage. And there we go. This is the chart that was produced. The bottom line is showing what's happening as the species is cooling, which is nothing very obvious. And the top line is showing what's happening as the species is warming, and you can see all sorts of interesting energy transitions happening there. The one that we're most interested in is here at minus 20 degrees Celsius, and you can see there's a major melting event happening at minus 20 degrees, which is the temperature that we thought we were safely storing our seeds at. As an example, a species that we know is long-lived at minus 20, so we survived, it's been in storage for 15 years. We pulled some out recently and it was still had very high germination. And so this is just showing the warming curve. Here's our storage temperature at minus 20 degrees and there's nothing much happening. So this is a good indication, a good way to see whether the seeds are likely to survive storage at minus 20. So here's 4 of our Myrtaceae species, and where that red line is, that's minus 20 degrees, and you can see there are major energy transitions either starting or happening around that storage temperature. So where we thought we were storing them at a nice in conditions that would make them nice and stable, they're actually being stored in a condition where the lipids inside are unstable. And that's the problem with these seeds is they're already dry, so it's not the water content, it's the lipids that are freezing and thawing that's creating the issue. The areas shaded in blue there are just showing areas below minus 100 degrees where there's nothing much happening, so that's an indication that maybe cryopreservation would be a better solution for these. But then we also have these areas for 3 species around minus 5 to 4 degrees that might be other alternative temperatures that we could try for storage. So we've done a bunch of storage experiments now to try and validate these results and see if we can find better temperatures to store the species at so they'll survive. And all the seeds except for 1 species have come from our potted collections and the living collections in the Botanic Garden because there's just no seed in the wild anymore. So those living collections provide a really good source of material for research, for providing seed that we can put into the bank, and then hopefully if we've got that material in the bank, then that's a good stable collection of material that we don't have to keep spraying and maintaining, so that we can just maintain that diversity while we figure out what else to do. So here's our first species. This is Archaeodomerta speculari. This one I made a good collection of back in 2015 when we first became aware that it was a problem, and so the seeds for this experiment were actually stored in the fridge for seven years before we put them through this process. So we used a moisture content of 8%, which is a little bit higher than we normally do, stored them for 4 months, and you can see already after 4 months we've got really poor response to storage at minus 20 degrees, but it's stored really well at minus 5 degrees. So that's giving us another option that we can use for storing those species. Lenwebia species main range, as Stephanie was saying yesterday, that's now thought of as a species complex with Lenwebia prominens and Lenwebia species black holes, so the black hole range, so the results that we've got for this are likely to apply to those species as well. So for this species, it's only been in storage for 4 months. We're now running another germination test where it's been in storage for 12 months, so fingers crossed they do as well. And so far there was no significant differences between the temperatures, but there was a bit of a decline as we were going into the colder temperatures. So I'm expecting with the next batch of seeds that we've sown that we'll see a big drop in survival at minus 20. But again, for this one, it's looking like 4, minus 5, and even minus 192 degrees is a good, or are good alternatives. Rhodamnia maydeniana, on the other hand, this one was moisture content 3%, stored for 12 months, and you can see already there's a really huge drop in viability for that species at minus 20 degrees, but it's doing particularly well at minus 5 degrees. So that was matching up with the profile that we had from the thermal analysis, which is indicating that thermal analysis is a good way to see a better temperature to store the seeds at. Rhodamnia rubescens, I expected to respond the same way as Rhodamnia maydeniana, but it hasn't, and so far it's doing pretty well in storage at whatever temperature we tested it at. Again, we've got another experiment running where they've been in storage for 12 months, and so I'm expecting survival to drop at minus 20 degrees. But for this species, we've got options at 4, minus 5, and minus 192 that are looking like they're good options for storing this species. And our last one, Syzygium anisatum. This is very unusual amongst the Syzygium genus in that it produces a dry fruit. But we found that with artificial ageing studies, it's likely to be really short-lived in storage at minus 20. So for this species, it didn't look like it would survive from the thermal analysis at minus 5 degrees. So we just tested minus 20 and minus 192, and it did really well at those temperatures. So that species is a good target for cryopreservation. So all the species look like they're storable, but the optimum temperature differs, and optimum moisture content also seems to differ, so it takes a little bit of work to figure out the right conditions. As a stopgap, we can store all of them at 4 degrees until we figure out the best lower temperature to store them at. And those other alternative temperatures are looking like there's something that we can find out with thermal analysis. So that looks like a good opportunity, a good outcome to let us preserve the species longer term without having all those maintenance costs associated with the living collections. That's it, and I'd just like to thank all those who've been funding the Rainforest Seed Conservation Project, particularly the Ian Potter Foundation, who funded this project on using thermal analysis, and all those who've contributed to laboratory and nursery assistance for the project. Thank you.
That was really interesting. So when you've frozen those seeds down, are they metabolic? Is there any metabolism going on in them, even at those really cold temperatures, or is everything stopped?
Theoretically, everything's stopped in cryopreservation, so at minus 192 degrees, there's nothing much going on. Yeah. But apparently there are some very, very low-level reactions happening. Yeah. Okay. Minus 20, there's a bit more happening.
Thanks.
I just had like a sort of a technical curiosity. There were those species that you had that were quite stable in the cryo sort of zone, less than negative 100 degrees, but were misbehaving at about 20. Is it not a problem when you sort of bring them back up through that range to warm them up again, at that zone, or is the crucial point that they're not stored for a long period of time at that?
Yeah, I think the thing with those species is that they will survive very short periods of storage at minus 20, so it's okay to bring them through it, but it's the longer term storage that's the problem. Yeah.
Any other questions? Yes, one.
So I was wondering, in the species we actually do see a drop down of getting them back, do you think that's actually genetics of the seeds, or do you think it's a random effect of basically it has nothing to do with genetics and all the seeds will behave the same, or do you think there's actually something in the species which leads to differences in being able to come back?
So you're talking about differences within a single species in response to storage?
Yeah.
Yeah. Yeah, that's a good question. I'd say there's definitely a genetic component to it. Between species, the differences are likely to be in the composition of the lipids, but within the species, yeah, it's a good question.
Kia ora. I was just wondering if you vacuum pack that, put it in with nitrogen to minimise the oxygen effect.
Yeah, that's our standard method for the seed banking. So we just vacuum seal them into those foil packets, which is really useful, really quick, really simple way to do it. So there was no impact from oxidation.
Awesome, thanks.
Okay.